Good afternoon, colleagues and students. It is our great honor to have the Shaw Laureates in Life Science and Medicine 2024, Dr. Swee THEIN and Professor Stuart ORKIN, with us today to share their award-winning research discoveries. The Shaw Prize Foundation was established in 2002 under Mr. Run Shaw, a visionary philanthropist who strongly believed in the power of knowledge. The inaugural Shaw Prize was presented in 2004. It gives out 3 awards every year: in astronomy, in life science and medicine, and in mathematical sciences. Regardless of race, nationality, gender, and religious belief, the Shaw Prize honors individuals who are active in their respective fields and who have recently achieved distinguished and significant advances, who have made outstanding contributions in academic and scientific research or applications, or who, in other domains, have achieved excellence. The Shaw Prize is dedicated to furthering societal progress, enhancing quality of life, and enriching humanity's spiritual civilization. HKUST takes pride as one of the partners, together with CUHK and HKU, to witness its success over the years. As we gather at this moment, I would like to thank the Shaw Foundation for bringing us 2 distinguished scientists to our campus. Please let me introduce our distinguished speakers. The first speaker is Dr. Swee THEIN. Dr. THEIN is the Senior Investigator and Chief of the Sickle Cell Branch at the National Heart, Lung, and Blood Institute, NIH, in the US. She earned her Doctor of Science from the University of Malaya back in 1999. After completing her specialist training in the United Kingdom, she served as Professor of Molecular Hematology and Consultant Hematologist at King's College London from year 2000 and 2015, while also serving as the Director of Red Cell Unit at King's College Hospital. Additionally, she led the Division of Gene and Cell-Based Therapy at King's College London from 2004 to 2010. Dr. THEIN joined the NIH in 2015 and is a Fellow of the Academy of Medical Sciences, United Kingdom, and Academy of Life Sciences for Chinese in the United Kingdom. The second speaker is Professor Stuart ORKIN. Professor ORKIN is the David G. Nathan Distinguished Professor of Pediatrics at Harvard Medical School, the US. He obtained an MD from Harvard Medical School in 1972. He held various academic positions at Harvard from 1978 to 2017, and has been the David G. Nathan Distinguished Professor since then. Additionally, he is an investigator at the Howard Hughes Medical Institute and a principal faculty member of the Harvard Stem Cell Institute. Professor ORKIN is a member of the US National Academy of Sciences, the American Academy of Arts and Sciences, and the American Philosophical Society. The topics of today's lecture are Stepping Stones to ECL11A by Dr. THEIN and Reactivation of Fetal Hemoglobin for Therapy by Professor ORKIN. Without further ado, please join me in giving a big applause to thank our 2 distinguished guests for sharing their fantastic scientific journey.
Good afternoon, everyone, and thank you very much for giving me this opportunity to share my journey in clinical genetics, leading to the discovery of BCL11A, which I entitled Stepping Stones to BCL11A. Just a little bit of background about myself. So I was born in Malaysia. I did my first medical degree in the University of Malaysia. I then left for the UK to pursue various training positions. After completing my internship and postgraduate training in internal medicine, I decided to specialize in hematology. And then in 1982, I made my way to Oxford and I joined the MRC Molecular Hematology Unit at the Weatherall Institute of Molecular Medicine in Oxford as an MRC clinical fellow. So David, who was head of the WIM or WIMM, was my supervisor, and he thought that a good project for me was to find out why some patients with beta thalassemia should have such a mild disease and do not need blood transfusions, unlike the majority who depend on lifelong transfusion. And then I was left to just find out this project and sink or swim. So, but whatever it was, this is where and when my interest and passion for the hemoglobin field began. And like good student, I did literature search. And it's actually been known for many years that patients with beta thalassemia and sickle cell disease, which is another inherited hemoglobin disorder that you will hear much more later, have milder disease when they produce higher levels of fetal hemoglobin. But it was not evident why the production of hemoglobin F was not suppressed in these patients compared to the majority of adults. And Janet Watson was probably the first person to make the correlation of the onset of sickle cell disease symptoms with the switch from fetal to adult hemoglobin. What she noticed was that the blood in the newborns with sickle cell anemia had very few sickle cells compared to their mothers, who also had sickle cell disease, where almost 100% of the red cells were sickle. And some follow-up studies suggest that this high hemoglobin F entity was inherited independently of the mutated beta globin genes. But this is where it was at that time. So I started then collecting families with mild beta thalassemia. And this is the outline of my presentation. For those who are not familiar in this field, I thought I would spend a little bit of time on fetal hemoglobin and what we mean by the switch from fetal to adult hemoglobin. And I'll walk you through the dissection of the genetic architecture underlying variability of hemoglobin F persistence. And from a genetics perspective, the story of hemoglobin F quantitative genetics uncannily mirrors the changing focus of human genome mapping, evolving from candidate genes through positional cloning to genome-wide association studies, or what we refer to as GWAS now. And I also then cover the key suppressor of hemoglobin F, which was uncovered to this study. So the hemoglobin switch refers to the progressive shutdown of fetal hemoglobin in parallel with increasing adult hemoglobin F production. So shown here is the sequence of human hemoglobin production during development. So all human hemoglobin is a tetrameric structure, is made up of two pairs of subunits, alpha-like and beta-like. The production of embryonic hemoglobin switches over to fetal, which remains the main hemoglobin during fetal life. And then just before birth, its production is steadily dialed down while that of the adult hemoglobin goes up. So then between 6 months to 12 months, adult hemoglobin is the dominant hemoglobin. And at this stage, mutations that affect the adult hemoglobin genes then become manifested. So when we talk about the switch, it really refers to the switch to fetal to hemoglobin, not embryonic to fetal. And when you look at the tetrameric structure, because alpha is part of fetal hemoglobin as well as adult, so this switch is really a switch from gamma to beta. And when adult hemoglobin becomes a prominent hemoglobin, which is by 1 year of age, the mutations affecting this adult beta globin gene become manifested. And the two main mutations are sickle cell disease and beta thalassemia. It is estimated that about 8 million people worldwide suffers from sickle cell disease, and this number is increasing. But together, the hemoglobin disorders affect about 20 million worldwide, the vast majority in low to middle income countries. So, what is sickle cell disease and what is beta thalassemia? Well, both are caused by mutations in the beta globin gene. But the disease manifestations couldn't be more different. So, for a start, all patients all over the world with sickle cell disease have the same mutation, which is a single base mutation in codon 6 of the adult beta globin gene, which switches transform glutamic acid to valine. And this abnormal hemoglobin, which is hemoglobin S or sickle hemoglobin, polymerizes under low oxygen conditions. And the hemoglobin S polymerization alters the structure and function of the red blood cells, causing them to adopt abnormal shapes, typically crescent or sickle shape. Hence the name sickle cell disease. These sickle cells are very stiff. They cannot negotiate to the fine blood vessels, causing obstruction, a blockage of the blood flow, and blocking oxygen delivery. And these are causing the acute clinical events, typically acute pain. At the same time, the sickle cells are highly fragile and they break up very easily, underlying the chronic hemolytic anemia that we see in sickle cell disease. And the two complications lead to multi-organ damage and early mortality. In contrast, beta thalassemia, although caused by mutations in the beta globin gene, is actually can be caused by a huge number of different mutations. I think today more than 300. The end result is the reduced production of beta globin, and it could either be no hemoglobin, which is beta 0, or just a small amount, what we call beta plus. So it's actually a quantitative disorder. And the pathology is actually caused by the continuing production of these alpha globin chains, which are highly unstable. And they precipitate out in the developing red cell precursors, causing premature death, which hematologists refer to as ineffective erythropoiesis. So when you look at the bone marrow of these individuals, there's a lot of erythropoietic activity, but it doesn't result in blood reaching the circulation. So that's why ineffective erythropoiesis. And this leads to excess iron absorption and anemia. So apart from sharing a common causative gene, they also manifest extreme variability in disease severity, even though it's caused by mutations in a single gene. And they also share two major ameliorating factors, one of which is fetal hemoglobin. And what is the mechanism of the ameliorating effect of hemoglobin F, or fetal hemoglobin? Well, in sickle cell disease, hemoglobin F decreases the hemoglobin S polymerization. You see here this tetrameric structure. So, because there is gamma globin in there from the fetal hemoglobin, you get a mixed hybrid, so you can have alpha 2, gamma, and beta S. And these do not participate in the polymerization process. So that's one way. And then secondly, the presence of hemoglobin F in the red blood cell dilutes out the concentration of hemoglobin S. And this slows the polymerization rate. Now, in beta thalassemia, the increased production of gamma will combine with excess alpha to form fetal hemoglobin. And this way, there's less unchaperoned alpha, and there will be less ineffective erythropoiesis. So I started collecting families, and two families, among others, convinced me that, beyond any doubt of the beneficial effect of hemoglobin F. So on the right panel here, your left panel, we see the patient who I pointed out the red arrow as TI, which is thalassemia intermediate or mild beta thalassemia. So this patient was really remarkable because we only knew he had beta 0 thalassemia when he presented for routine blood check prior to hernia repair. And he was 65 at that time, with no adult hemoglobin, all with hemoglobin F, and it's almost like 11 to 12 grams. And then we show here his wife, two sons, and an Italian colleague who accompanied me to collect the blood samples, and his granddaughter. So when I studied the rest of the family, I also found some of them had higher than usual fetal hemoglobin, even though they don't have beta thalassemia. And in the right panel, is another remarkable family. I was consulted about the patient which arrowed in blue, TM, thalassemia major. He needed regular blood transfusions of survival every 4 weeks. And when I visited the family, I found he had two other brothers, which is TI. Both actually had beta 0 like the brother, but they were not known to their primary care physician. And they had hemoglobin about 9 grams, all fetal hemoglobin, even though they inherited the same beta thalassemia genes like the brother with a severe transfusion-dependent form. So these two families tell me something. First, the hemoglobin F can compensate for the lack of adult hemoglobin. And then this ability to produce hemoglobin F is inherited separately of the beta globin complex. But what puzzled me was that in these two families, there were family members without beta thalassemia, and there were variable hemoglobin F levels. So I then thought maybe I should look at kind of normal people and collected healthy blood donors and just did the hemoglobin F levels. And shown here is the distribution of hemoglobin F levels, which has actually been studied by other people before me. And what you can see is that it's a continuous distribution and highly skewed to the right. So at one end, we have those with very high, which is actually not shown here, but they're rare, and they can have hemoglobin F as high as like 40%. And these turn out to be the so-called very rare Mendelian forms, which are inherited as alleles of the beta globin complex. The majority will have hemoglobin F of like less than 1%, and these residual levels of hemoglobin F is actually contained in a subset of erythrocytes, which we call F cells. I also then had to learn to develop assays for measuring these hemoglobin F levels in healthy people, because hemoglobin, the material, the equipment available then was not sensitive enough to measure the fetal hemoglobin in healthy people. So the levels of fetal hemoglobin in healthy adults can vary up to 24. So all of us in this room here will have some levels of fetal hemoglobin, and it can vary from 0.4% up to like 3%. But much more common is what we call the common variation. And these increases are modest, can be up to like 4%. And it is also known as hectocellular because the small levels of fetal hemoglobin are unevenly distributed. And when you have this common variation, you can see that it is actually amplified when the patients have sickle cell disease or beta thalassemia, which is also highly skewed. So above is the normal distribution we see in healthy adults, but below is a number of patients with sickle cell anemia. And you can see, so they don't have the HPFH or the Mendelian forms, but the fetal hemoglobin levels can vary from like 1% right up to like 25%. So I've shown here a classification of the primary inherited conditions, because hemoglobin F levels can also be increased as secondary to some conditions when there is acquired disorders, where the increase is due more to perturbations in the normal process of erythropoiesis, like in pregnancy. But this shown here is the primary inherited conditions with increased levels of hemoglobin F, or what we refer to as HPFH, or hereditary persistence of fetal hemoglobin. And I've classified them here molecularly, those caused by deletions varying lengths involving the beta globin complex, and the non-deletion type. And within the non-deletion, those which are linked to the beta globin complex, which is generally caused by point mutations in the gamma promoter. So the linked forms, the deletion, and those caused by the gamma promoter variants, follow a Mendelian inheritance as alleles of the beta globin complex. And you can recognize the carriers easily because they will have definite increase in fetal hemoglobin from 15 to as high as 35 or 40%. And the F levels are distributed evenly, what we refer to as pancellular. And here, what I would like to show you is that at that time, it's a very old slide when I was actually studying this, studying in the hemoglobin field. And these point mutations are really clustered in two areas. I'll come back to this slide a bit later, around -100 and around -200. And it seems probable that these mutations are sites for potential binding sites for transcription factors. The question is, which ones? And there was a lot of activity in this area then, all trying to understand the switch. But uncovering the transcriptional repressor was elusive. So then let me come back then to the other form, which is a common hemoglobin F variation or hectocellular. So these actually do not follow a Mendelian inheritance. The inheritance pattern is complex. And just to a few points here, it is not a distinct genetic condition, but really an extreme expression of a quantitative trait. So you can think of it as a combined expression of multifactorial discrete traits. So it is really a phenotypic classification. with modest increases in hemoglobin F of less than 5%. And unlike the Mendelian forms, it's quite common in the normal population, it's about 10 to 15%. There's no clinical importance per se, but when co-inherited with beta thalassemia or sickle cell disease, it leads to high hemoglobin F production with major beneficial effect. And of course, the beta globin complex is intact. And family studies also show that these high F levels tend to be inherited, but the pattern of inheritance and the strength of genetic component was not clear. Having said that, the first kind of QTL for this common variation is probably the XMN1 gigamocyte. So this was like a candidate gene, if you like. People who were studying sickle cell disease, the clinicians and also epidemiological studies noted that those who had the hemoglobin S inherited on the Senegal, then, at that time was very popular to actually type the sickle cell disease, the haplotypes. So those who had the hemoglobin S mutation on the Senegal beta S haplotype or the Arab Indian tend to have high fetal hemoglobins and a milder disease compared to the other two, the Benin or the Bantu. And what they have in common, the mild ones, was the T allele of the XMN1 gigamocyte, which was also present in those with the common variation or the heterocellular HbFH. So although the presence of the XMN was associated with these modest increases in hemoglobin F in sickle cell disease and beta thalassemia, it was modest. So it didn't explain all the very mild forms. So then I thought I should have to maybe try to attack the genetics of this complex hemoglobin F variation. And I used kind of two approaches. One was population studies based on twin and SIP pair analysis. And the second one was linkage analysis in a single extended pedigree. And just I was very fortunate at that time when I was doing this study to come across Tim Spector, who was director of the Twin Center then. And Tim was actually collecting twins, identical and non-identical, for various studies. And I said to Tim, I said, I would like to see how well hemoglobin F levels are inherited. But of course, we don't measure hemoglobin F levels then because we still don't. It's part of a routine CBC. So each week we might make 1 or 2 trips down to the twin center and collect blood samples from these twins. And the first study was just to see how well they correlate, in identical and non-identical twins. And twin studies are the kind of traditional way to actually find out about the genetic component, like what people do for schizophrenia and other diseases. What I can show here is that in the monozygotic twins, when the F cell levels of twin 1 is on the y-axis and the twin 2 is on the x-axis, and you can see they're highly correlated, almost in a straight line, compared to that of dizygotic twins versus random scatter. And then in parallel, I also looked at the inheritance patterns in other red blood cell variables. And you can see that the relative proportions of the additive genetic component or the heritability in F cells is 0.89, and showing that F cell levels are overwhelmingly genetically controlled compared to the other red cell traits. So this actually gave me a lot of confidence that it was worth changing the genetic traits or the genes underlying this common hemoglobin F variation. So the question then is this large genetic effect, and it was British European twins, sampled due to one of few major loci, or is it polygenic? And what is the effect from the beta globin complex and the XMM1G gamma site. And when we did a SIP pair analysis using 316 same-sex, non-identical twins, and we did this to remove the confounding effect of the hemoglobinopathies, age and sex. And we found that more than 50% of the F cell variants remain unaccounted for. After we accounting for the beta globin complex, most of it which was due to XMN1G gamma site. And 50% of this unaccounted for was trans because it's not linked to the beta globin complex. So the next step then was to how to dissect the complexity of this hemoglobin F and F cell inheritance. And fortunately, I was able to come across this Asian Indian family, and we extended it to a huge kindred. But just to come back to this about linkage analysis. Linkage analysis actually aims to establish linkage between genes that co-segregate with a trait or disease within a family. And because it is within the family, we need to have informative genetic markers to help identify the genes close to each other. And evidence of biological relationship is really important. So I said to David, I said, I've got this Indian family and I really want to try to extend it. And part of this family was in Malawi. So he said, okay, off you go then to Malawi. So I did make this field trip to Malawi and managed to extend this family to 210 members, spanning 7 generations. Of course, we didn't manage to collect blood samples way back to the first generation. But we managed to collect quite a lot, which is about 200 10 family members. Then, if you remember, I need to prove, biological relationship. And here, again, I was actually very fortunate because I came across Sir Alec Jeffreys, who developed these polymorphic probes, which are using mini satellites. And this family, which was a lot of marriages within the family, was really ideal for Alec because he could show that they were so polymorphic, and every one of these family members had a unique DNA profile, leading him to coin these probes as DNA fingerprints, which is now used extensively in many studies, including solving paternity dispute. So with this, I was confident that there was evidence of good biological relationship within this family. Then, of course, the next thing was needing polymorphic probes. And here I was also very fortunate to, come across Mark Latrobe, who is a Canadian biostatistician who was then working at Oxford. And Mark had developed these microsatellites, which are dinucleotides, and which seems kind of perfect for this very consanguineous family. And we accounted for the XMN1G gamma effect and beta thalassemia. And we could then localize the gene, if you like, that was responsible for high F in this Indian family. And apologies, I forgot to mention that in this family, the proband of the patient also had very mild beta 0 thalassemia and could show that it was inherited independently of the beta globin complex. So at this stage, within that family, we could map this, what I call the second hemoglobin FQTL on 6q. And within this huge interval of 1.5 megabase, we found 5 protein coding genes. We sequenced all of these. We found no mutations, but 3 of these genes were expressed in the rich progenitor cells. And we decided to concentrate on 2, which is HKUST and MIP, because both are involved, could be involved in hematopoiesis, but certainly MIP is essential for rich differentiation. But this interval is still far too big. So the question is, how can we narrow this 1.5 megabase 6Q interval? It was no longer possible within this huge family because they were all linked, and we'd taken it as far as we could. And we decided to then check if it was present in the and we then decided to see if it was present in those British European twins, because you remember I happened to have the samples. And in fact, to our delight, they actually found that those British European twins also, the high F were also associated with this interval. And we used two panels, and we could localize it to an intergenic interval between HBS1L and MIP. And here we actually sequence this whole interval and identify 176 single nucleotide polymorphisms or SNPs. And we could show that these 28 of these SNPs provided very strong evidence of association in two panels of the DZ twins which we used. And if we use MZ or monozygotic, we use only one for obvious reasons, because the other one will have identical genetic makeup. And then we found that these 28 SNPs could be narrowed in 3 blocks. The huge linkers disequilibrium blocks. And the one in the middle, the HIM or HBS1L MIP intergenic polymorphism 2 had the block of highest association. And what you could see is on the left, it was associated with high F cells, particularly the block 2, and this effect was seen more in the Asian Indian family than the Caucasian family. And the block 2 variants were also associated with hemoglobin F levels in patients with sickle cell disease. The next question is, how many hemoglobin F QTLs are there, and how do we find them? So at this stage, we could now do a genome scan using SNPs. And one of the earliest one was actually, there were like 300 SNPs available made by Illumina. And Mark and I thought, well, we have more than 5,000 twins. We do not have the resources, and it would take a lot of time to actually genome scan all the 5,000. And then came up with an idea. We said, why don't we just look at the extremes? So we took those with F cells less than the 5th percentile, and then the other one, which is more than 95th percentile. You'll see that this is a bell-shaped curve because we log transformed the F cells. And then we did genome scan on this. We narrowed it down to 179 individuals. And to our surprise, we actually found, first we confirmed the site on the beta globin complex, which is on 11p. And then we also confirmed one on 6q23, which is the interval between HBS1L and MIP. And we found this other one on chromosome 2p. And that turned out to be BCL11A, which was a zinc finger protein. And the SNPs, which are associated in this region, were found in 2 clusters. The highest one was actually in intron 2, which I've arrowed here. And this will be, Stu will probably talk more about from where we take off from this localization to bringing it to fruition for therapy. So a note here, BCL11A was not known to be involved in erythropoiesis until this genome-wide scan, but it has previously been implicated in neurological development and myeloid leukemia and lymphoma, but not in erythropoiesis. And then following this, there were reports of naturally occurring rare BCL11A variants in patients with autism-like neurological syndrome. And they were associated with very high hemoglobin F levels, implicating the importance of BCL11A in erythropoiesis. Now bring it back to this slide. Remember I showed you that those gamma promoter mutations were clustered in two areas. And in 2018, two groups, one by Stu's group, showed that in fact BCL11A actually binds to the cognate sites around -100, which then tells us that if you can disable binding of BCL11A or disrupt those cognate sequences, it could actually recapitulate those naturally occurring HbFH. Now we come back to 6q. So how does this HIM2 locus on 6q influence hemoglobin F levels? Our hypothesis was that this region contains regulatory elements controlling MIP and or HbS on S1L expression, which in turn influence hemoglobin F and other erythroid traits. We did some transcriptomic studies where we did erythroid cultures of people with high F versus low F. And we could show that those with high F individuals actually have lower CMIP expression. There less erythroid differentiation, erythroid proliferation, but the erythroid maturation is accelerated with higher number of macrophages. And we could prove this genetically too. We showed that those with the hemoglobin F boosting allele in this HMIP2 region were associated with homozygous for this site compared to the wild type, which is 1-1, actually had lower red cell count, higher MCH, higher MCV, higher platelet count, and lower levels of monocytes. And this association with the erythroid phenotypes has been replicated and validated in populations of diverse ethnic background in several meta-analysis studies. So this was our proposed model for the effect of HIM2 SNPs on MYC regulation. So we have those core intergenic variants, those in the HIM2 block. They affect transcription factor binding and reduce MYC transcription, which then, in the left panel here, this lower the MYC expression with less proliferation, fewer erythroid cells, but erythroid differentiation accelerated with effects on F cell numbers and red cell traits. And this is a plausible, what I propose, models of MYP and BCL11A mediated modulation of hemoglobin F levels. So here is the normal situation. In BCL11A, it probably has a direct effect on gamma globin expression by binding to those sites. And here, the F cell levels are higher, the hemoglobin F levels are higher, and it is evenly distributed. Whereas the effect from 6q is more like that of stress erythropoiesis, where erythroid maturation is accelerated, so bringing forward those younger precursors which are still synthesizing hemoglobin F. And these are the three major hemoglobin F loci. All three loci have been shown to be present in diverse ethnic populations with or without beta thalassemia. And in sickle cell disease, the QTLs were associated with reduced pain rate and also reduced organ damage. In beta thalassemia, they can predict disease severity and transfusion needs. This has been shown in Sardinia. And all the three QTLs were also associated with disease severity, hemoglobin F levels in patients with beta thalassemia and beta thalassemia in Thailand. And I think that the remaining variance in hemoglobin F is now due to polygenes, which contribute very small effects and or rare variants. For example, KLF1 and DNMT1. And with this, I would like to acknowledge the contributors and the people who have really contributed to this almost 20 years of work. So Alec, who actually helped me in the very early part of the study with the single Asian Indian family. Well, Tim Spector came along at the right time when I needed some confidence to actually change those genes. And Mark was also, I was very fortunate to know Mark at the right time, who came along with his polymorphic probes and then helped me tease out all those hemoglobin F loci. And Jamie was my stalwart PhD fellow, and we churned through lots of polyacrylamide gels while we were running those microsatellites. And Stefan was with me in the last lab when we led to localization of BCL11A gene. And with that, I would like to thank you.
Thank you very much. I'm really pleased to be here and honored to present as part of this Shaw Lectures. And what I'll do in my talk is, in a way, continue the story that Swee presented. And in the beginning, there'll be a few slides you'll see that will be very similar or maybe even the same panels, which I think will just sort of emphasize some of the background material. I'll then tell you how we got to an editing therapy and how we applied the knowledge regarding the mechanism of the hemoglobin switch. And then in the last portion, I want to describe some newer data, which I think is quite interesting and suggests ways that we may even develop additional therapies based on mechanism. So I think one message that will come through from this talk is that we've really entered a new era of molecular medicine now, where new technologies are being brought to patients, and in some cases now via stem cells. And I think an example would be editing of cells as well as CAR T-cell therapy, for example. And I think the best example of that is the approval last year of the first gene editing therapy, which is known as Casgevy, which was approved in many jurisdictions and was brought into clinical trial by CRISPR Therapeutics and Vertex Pharmaceuticals, based on our work and licensed actually from my institution, Children's Hospital. But I came at the problem differently than Swee perhaps. My interest was originally in sort of fundamental aspects of blood cell development. Where do blood cells come from? How do they form? What might be mutations when one has cells that go awry? And how can we develop new therapies? And I chose the red cell in part because it's a relatively simple cell. We knew quite a bit about it. And there's a long history now in molecular science using hemoglobin really as a platform in red cells. And I like this quote from the great geneticist Arno Matulsky 50 years ago, where he said hemoglobin research plays a role in human biochemical genetics similar to that of Drosophila research in formal genetics. And I think if one traces the history of molecular biology, cloning, gene expression, genetics, editing, hemoglobin has played a part in almost every one of those subjects and advances. Now this is a very similar representation of what Swee has shown, the switch from gamma to beta gene expression, which is a transcriptional switch in the beta cluster on chromosome 11. And in the adult, we all have a small trace of fetal hemoglobin, about 1%. And there are two major disorders, again, sickle cell disease and beta thalassemia. In the 1980s, we worked through the mutations in beta thalassemia with the hope of understanding how red cells are formed and how gene expression is programmed in those cells. But in fact, the discovery of the mutations was more valuable for prenatal diagnosis than understanding the red cell. And sickle cell disease has a prominent history in human genetics as the first molecular disease, is coined by Linus Pauling, and then Vernon Ingram discovered the amino acid substitution 7 years later. I think it's important to recognize, though, that knowing the molecular basis of sickle cell disease and also thalassemia really has had, until recently, little impact for the patients. The therapies have not relied on any understanding of the molecular basis. And this is a classical patient with beta thalassemia. And again, I think using a panel that Swee had shown, just to emphasize, these are not rare disorders. They're global disorders with 300,000 new births per year for sickle cell disease, mostly in resource-limited areas of the world. And again, we've known for many years that hemoglobin F is protective, both in sickle cell disease and beta thalassemia, from the astute clinical observations of Janet Watson that Swee mentioned, and also the natural history study done through the NIH about 30 years ago, a little more than 30 years ago, showed that higher level of fetal hemoglobin led to better survival of patients with sickle cell disease. And the sickle hemoglobin, fetal hemoglobin differs from adult hemoglobin, having a slightly higher oxygen affinity, which is really not a major consequence in the sense that individuals with 100% fetal hemoglobin through very rare mutations are perfectly normal. And the hemoglobin F can effectively replace hemoglobin A, and F has the benefit of blocking the sickle polymerization. And as Swee has shown, hemoglobin F is under genetic control, and the persistence of fetal hemoglobin has two genetic bases, one common variation and one rare variation. So the therapeutic vision in the field for 50 years actually has been to increase the level of fetal hemoglobin in the adult from that 1% to maybe 20 or 30% as a therapy for either sickle cell disease or beta thalassemia. But the challenge is that we didn't really understand how this switch is controlled. And this is really the crux of the problem. The locus control region, which is a super enhancer upstream of the coding genes, loops to the downstream globin genes to activate their expression. And at the fetal liver stage, the duplicate gamma genes are activated by the LCR. And in adult bone marrow, this is prevented, and the beta gene is activated largely as a default. And what we didn't know was how many factors control this switch, what the factors were, did they act positively or negatively. If there were many factors, this would make it very difficult to manipulate them in a therapeutic sense. If there are a limited number of factors, one would have a better opportunity. The first factor we actually isolated to try to get to this problem was the transcription factor GATA1, which we isolated back in 1989 at the same time as Gary Felsenfeld. And this turned out not to be a regulator of fetal hemoglobin, but really a master regulator for all of erythroid gene expression, and founded a whole family of GATA factors. But GATA1 has no stage specificity. It's expressed in embryonic, fetal, and adult stages, and therefore cannot explain the globin switch. And so if we come up to that period in the 1990s, we understood a master regulator, but didn't understand what controlled the switch. And for a long period of time, the field was kind of wandering in the desert trying to identify these factors. The path to B7A, which Swee defined, really depended on genetics. And this is the path that we've taken subsequent to that. We collaborated independently with a group in Sardinia that was doing a GWAS at the same time, which led to the same associations you described. And then subsequent to that, we worked on the cell biology, genetics, and the mechanism by which B11A represses fetal hemoglobin, and also on applications for therapy. The challenge in genome-wide association is one looks at the entire genome of 3 billion base pairs and has to identify a particular trait and then ask, how does that hit in the genome, that GWAS, really relate to either the mechanism of that trait or a particular disease? And in most cases, this is unresolved. Most instances of GWAS associations in the literature go without a mechanistic basis for what the association is. This was a summary of the GWAS, in which we collaborated with the group of Antonio Cao in Sardinia at the time, same period of time as Swee described, which identified B11A, the HBS-L1 MIB region, and the human beta clusters associations. The first evidence we had that B11A actually might control fetal hemoglobin is this relatively straightforward experiment in which we use CD34 progenitor cells, which are the same cells used in bone marrow transplant. And we could differentiate these cells in culture to erythroid cells and use shRNA to inhibit B11A expression and ask what happens to gamma gene expression in fetal hemoglobin. And an effective shRNA leads to an increase of gamma gene expression and fetal hemoglobin. And so this was the first experiment which suggested a direct connection, perhaps, between B11A and hemoglobin F. Although in retrospect, I think we were lucky because there are many, many things one could do to these cells to get a change in fetal hemoglobin. So this is not really proof, but I think it's a suggestion. A more convincing experiment is this one, which was performed a couple years later, in which we asked how important is B11A as a single factor. And in this case, we used transgenic mice, which are called sickle cell mice. These have the mouse beta genes eliminated and the human gene inserted instead. They were made many years ago by a couple of groups. And if one introduces the sickle human gene, one has a mouse that essentially has sickle cell disease. We interbred these mice with B11A conditional mice over here and used now sort of classical gene inactivation using Cre recombinase to remove B11A in the erythroid lineage alone in order to avoid any confounding effects in other cell types. And the question was, if we remove B11A, what would happen to the sickle cell phenotype of these mice? And what was remarkable was that they were sickle cell phenotype was completely abolished, avoided. Here's the mouse with sickle cell disease, and the interbred mice had a normal smear. They have normal hematology. They have high level of fetal hemoglobin. And this experiment showed that manipulation of just one gene is sufficient to reverse the sickle cell phenotype. This experiment was done 13 years ago now, 2011, and it's the rationale for virtually all of the genetic trials that are being done today. So knowing that this was a critical factor, then we could add this to our sort of simple diagram and show that B11A is able to repress gamma gene expression and promote the switch. Beyond that, B11A is actually a continuous quantitative regulator. The system is completely hardwired. For the most part, if you know the level of B11A, you know the level of fetal hemoglobin production by those cells. And really an example of that was sort of alluded to by Swee in this syndrome, an unfortunate syndrome of haploinsufficiency, which is an intellectual disability syndrome that occurs through either deletion or mutation of one copy of B11A. These individuals have an autistic-like phenotype, but really the issue for us was what's their level of fetal hemoglobin, which was the least important thing for these unfortunate children. But what we learned by looking at about a dozen of these individuals is that their level of fetal hemoglobin is approximately 10 times normal in the range of 13 to 15%, which is really in the sort of classical range of what's called hereditary persistence of fetal hemoglobin. And this is from having one copy gone, so essentially 50%. So this told us that if we wanted to use B11 as a target for therapy, it's not necessary to ablate expression. We could reduce its expression to raise fetal hemoglobin, which might provide a sort of measure of safety, if you will. But the challenge was still, how did the GWAS originally impact the B11A gene or locus? We knew that the protein was the same. It's a hyperconserved coding region gene, so it had to affect the regulation of B11A itself. And Dan Bauer, who was then a fellow at the time, really took on this problem. And I showed that the most highly associated SNPs were sort of overlapping 3 DNase hypersensitive sites in erythroid chromatin, and this, which is not, these sites are not present in either B cells or brain, which are also cells that express B11A. And that's really the hallmark of a regulatory element or an enhancer. And blowing this region up, it's about 10 to 13 kilobases in length, 3 DNase hypersensitive sites. We then took that region and introduced it with a reporter into transgenic mice and showed that it's a erythroid-specific and adult-specific enhancer. And this cell specificity is really key to the editing therapy that's been developed, because any modification in this region of the gene, this regulatory element, is only has a consequence in erythroid cells, has no consequence in brain or B cells, where the protein is also important. Additionally, we showed this enhancer is absolutely essential for B11A expression. If you remove this enhancer, B11A is not expressed in the erythroid lineage, but is expressed normally elsewhere. We could actually make mice that lacked the enhancer, had slightly perturbed hemoglobin expression, but otherwise these mice are perfectly normal. The 10 to 13 kilobases was too big a region to think about genetically modifying for any form of therapy. But we also wanted to understand sort of the structure of this enhancer. And fortunately, about the same time now, which is 2015, CRISPR came on the scene and allowed us to essentially do a saturation mutagenesis of this region, really a unbiased mutagenesis, and asked which portions of the enhancer are required for B11A expression. And the remarkable finding of that was that there's a single site, I call the Achilles heel, in the middle DNase hypersensitive site. Here's the guide RNA directing the CRISPR. And this site is a GATA transcription factor binding site, so it hits the binding site for the major erythroid factor. This single site accounts for probably 2/3 to 3/4 of the entire function of this enhancer. So a single cut in 13 kilobases is enough to cripple the enhancer functionally, although not completely. And this was obviously of tremendous implication therapeutically. And this guide RNA is precisely the guide RNA which is used in the editing therapy. This guide was reported in 2015 in our paper. And this, the fact that common variation impacts an enhancer regulatory element is a common theme now in genetics, that most of the common variation occurs within regulatory elements and not coding region. We then began to focus on BCL11A itself. What is the protein? How does it actually function? The BCL11A protein, which I'll come to a little more depth later, it's about 100 kilodaltons in size, and has 7 zinc fingers, but the 3 terminal zinc fingers, called 4, 5, and 6, bind a unique sequence called a TGA-CCA, which was not present in the database previously. And here's a structure of it, structure of the protein, this region on DNA. This sequence is remarkably present in the gamma promoter at the minus 110 to minus 115 region, which Swee mentioned. I'll come back to that. And what we've shown through a variety of studies is that BCL11A binds the sequence TGA-CCA, which is present in the gamma promoter. It displaces the activator NFY, which binds a little further over here, recruits the NERD co-repression complex, closes the locus to make it essentially inaccessible, represses the gamma gene, which then allows the beta gene to be expressed. And this is only one of 2 repressors. The other repressor is LRF, or is DBTB7A. It binds at the minus 200 region. And that protein also interacts with the NERD co-repression complex. So there are 2 repressors, about equally potent, but removing the binding site for either one is sufficient to reactivate gamma globin gene expression. And there's competition in the locus with NFY, this activator. Soon after we reported the results with the enhancer, CRISPR Therapeutics, a new biotech company in the Boston area, took up the challenge of translating this to the clinic using a guide RNA that I showed you before. And relatively rapidly, they reported in the first clinical trials 2 patients. One patient with severe beta thalassemia, and another patient with sickle cell disease. And in both of these patients, editing at the Achilles heel, as shown here, led to essentially functional cure of these individuals, such that the individual with beta thalassemia no longer required transfusions, and the patient with sickle cell disease no longer had painful crises. And they continue these trials in collaboration then with Vertex Pharmaceuticals, leading ultimately to the approval. But I think it's important to recognize the impact on the patients and their lives. This is actually the first patient who received the editing therapy, Victoria Gray, who's been quite prominent in a number of meetings. And she just describes that before the therapy, her life was miserable. She couldn't look to the future, and after the therapy, she now has really a full life ahead of her. And this has been the experience of most of the patients. So what is the therapy? I think many people don't really realize what's involved. The therapy involves first harvesting what's called mobilizing stem cells from the blood of an individual. These are then collected, taken to the laboratory, and modified. In the editing therapy, they're modified at the Achilles heel with CRISPR-Cas9. Once one has the modified cells, one preconditions the patient. What that means is the patient host bone marrow is destroyed, most typically with busulfan, a chemotherapy agent, and then the cells are re-administered to that patient, and the hematopoietic system sets up again, and the patient is then has been treated. The whole process takes 6 to 7 months minimum, It takes tremendous commitment on the parts of the patient, complex resources, which really can't be delivered in many places. But the therapy, the results of the trial were really quite impressive. These are the results presented to the FDA, the United States Regulatory Agency, which this is for sickle cell disease, where the black dots are hospitalizations for painful crises prior to therapy, and after the therapy, there are only a couple of patients who have had problems thereafter. The results then are transformative for these individuals, and the same has been true in patients with beta thalassemia, where 95% of the patients essentially have been rendered free of transfusions. I think the obvious problem is we have a global disease where we have thousands of patients, and we have a procedure for cure that's essentially one patient at a time, a bone marrow transplant. Before sort of moving on, I just want to reemphasize some of the benefits, perhaps, of increasing fetal hemoglobin as a therapeutic approach. One is, I think it's thoroughly validated by genetics, as Swee has shown, and it's also validated by the clinical trials. It maintains the proper balance of globins. The alpha and beta globins have to be in the right ratio with respect to one another. And in the other now approved therapy, which is a lentiviral globin addition therapy, where beta globin is expressed from a lentivirus, that balance is not maintained. Whether that's clinically important in the therapeutic setting like this, we don't know. The important thing is this is suitable for all hemoglobinopathies, for sickle cell disease, and all of the beta thalassemia mutations. So it's essentially one-stop shopping. And now we know there are many different routes to increasing fetal hemoglobin. The first one is cutting the enhancer. But there are other routes. My colleague, David Williams at Children's Hospital, has used shRNA to downregulate B11A in the context of a lentivirus. But perhaps more in the future is modifying the binding site for B11A in the gamma promoter, which is under study right now. And each one of these has a sound therapeutic basis. And in case of sickle cell disease, it also obviates, usually people ask, well, why don't you just correct the sickle mutation? Why do you go to all this trouble to raise fetal hemoglobin if we know the sickle mutation? Well, the problem is technically correcting one base is much more difficult with gene editing, even today, than just cutting the DNA. And this avoids those issues with respect to sickle cell disease. Just want to give you a sense of where the clinical trials are. There was a clinical trial initiated to correct the mutation in the sickle gene using homologous directed repair. The first patient had unfortunate events in terms of reconstitution of the blood system, and that trial was terminated, and the company stopped the program. This is a company, Graphite, in California, showing really how challenging doing these clinical trials really is. In terms of B11A-directed therapy, there's downregulation of the RNA with shRNA, which is into phase 2. There's approved therapy of Casgevy using CRISPR to cut the enhancer. And then there are two other therapies now directed to the binding site for B11A and the gamma promoter. One by company Editas in Cambridge, which is using a different editing enzyme. That therapy has been used for several patients, but the company about a week ago said they're seeking a partner. They're not going to go further by themselves. It's very expensive to do these trials, and obviously there's a competitive landscape. And another company, Beam Therapeutics, is using base editing, where it doesn't cut the DNA, which is theoretically perhaps safer approach. And they're also using that on the promoter because there's one can use a C to T conversion there. So that's the current state of work. I think there will be second and third generation editing therapies coming to approval, but it may depend on the financial viability of these for ex vivo therapy, which is challenging. So in terms of the timeline, we've had a long history in this field, starting back in 1949, culminating, I think, in the approval of the editing therapy. So there are some limitations now to genetic therapy, as I've said. We know it works, but it requires tremendous resources. It's clinically involved, it's expensive. One has to precondition the patients, which carries toxicity. It's very low throughput. And so how can we do better? Well, we need to increase the throughput, if you will. And one way to do that is to make the preconditioning less toxic. And there are many laboratories and biopharma that's involved in trying to do that. Another approach is to use in vivo genetic therapy, and that is inject something like a lipid nanoparticle with RNA that might be modified. And that's under study as well. I think one of the challenges there is one needs to modify a significant fraction of stem cells to get in the therapeutic range of the hemoglobin disorders, probably 50% or more. And I think that's quite challenging with current technology. So a number of us think we need a pill. We know how to, it seems kind of retro, I think, but we know how to do that. We know how to give people pills, and we know how to send pills to other parts of the world. And I think one sort of way to bring that home is there have been several articles published, for example, in the New York Times, indicating really how challenging it is for patients to not only get into these trials, but also to manage to get through the whole trial. It's a long process. So the scope of the problem, I think, is currently practiced, the gene editing or gene therapy that we have cannot reduce the burden of disease, even in resource-rich areas of the world. And in vivo gene therapy, I think, is still a vision, and we don't know if it's possible. The standard of care drug for sickle cell disease, hydroxyurea, is beneficial, but only for a fraction of patients, and doesn't forestall the complications. And investment in pharma, by pharma, philanthropy, the NIH, really has been inadequate, I would say, to the challenge given the number of individuals who might benefit. And so what we're trying to do in our own group is to leverage basic findings for novel drug discovery. And I'll take the last few minutes to describe sort of why I think that may be possible with recent findings. B-cell has a transcription factor, and transcription factors have always been viewed as challenging and undruggable by pharmaceutical companies, which has led to the ways to circumvent this undruggability, using looking for interacting proteins, looking for molecular glues, for example, other dependencies, and to target these rather than the protein itself. And so the approaches that have been used in the field sort of are historically the approach that's been used to find agents that would raise fetal hemoglobin is to take cells, essentially take the kitchen sink, whatever you have on hand, add it to the cells, and try to find things that induce fetal hemoglobin. That, I'd say, has been universally a failure, in part because these cells respond to many, many things that will change their differentiation, growth, et cetera, that give you a readout. Another is to look at the mechanism by which switching occurs and to try to focus on cofactors or epigenetic factors. I think this is a little more on target but still is less potent and less specific. And so, I would argue that once we know that the proteins are involved, target those proteins, because we know what the effect will be. They're the most potent, but they're also the most challenging, which is really the challenge. We're trying now to leverage our understanding of the mechanism to inform discovery of targeted agents. And I love this quote by Marcel Proust, the French novelist: "The only real voyage of discovery consists not in seeking new landscapes, but in having new eyes, looking at the problem differently." So the last few minutes, I'll show you how we're looking at the problem differently. The B11A protein is big. It's largely disordered, which is a bad thing if you want to find small molecules. The C-terminus binds DNA. The N-terminus binds the co-repressor complex. And it has these little zinc fingers sort of interspersed in this sea of disordered protein. If you go to AlphaFold and pull up B11A, it looks like spaghetti with a few conserved regions. It's not very helpful. We did have a hint that zinc finger 0, which is the orange zinc finger near the N-terminus of the protein, is involved in protein interactions. And so one of the experiments we've done is to remove that zinc finger in both copies of B11A in an erythroid cell line that is now sort of the standard in the field. And if one does that, there's no stable protein in the cells right here. So telling us that finger is essential. What was totally unexpected was when we isolate this finger as a recombinant protein, it forms a tetramer. And this is a sedimentation showing it's essentially a pure tetramer. We're able to crystallize this tetramer and determine its structure, and it's a symmetric tetramer with a hydrophobic interface. And it's really quite stable, has a dissociation constant in the nanomolar range. This is something that I've never heard of, a zinc finger forming a tetramer, but that's sort of the way it's come out. The next finger down, zinc finger 1, is another small finger. These are only 20 some odd amino acids. And it has in the red highlighted section hydrophobic amino acids that resemble a degron, a protein degradation signal. So we thought, well, maybe this is important too. And we removed this finger, again with CRISPR editing in these cells, both copies. And what we find is, what we find is there's more protein than the wild type. Remarkably, this finger forms a dimer if one isolates it as a recombinant protein. And this is actually the structure of it predicted by AlphaFold, but we actually have the protein structure now, which agrees quite closely. So we have an unusual setting in which we can remove one domain of a protein and get essentially no protein produced at steady state. And we move another finger, we get more protein. How do we make sense of this? Well, I won't go through all the data. I'll just tell you what we think is the answer. And it's shown here, is that when the protein is first expressed, we think on the ribosome, there's zinc finger 0, zinc finger 1, and you can imagine more protein over here. This degron sequence is exposed, and the protein is destroyed and goes to the proteasome. At the same time, or thereabouts, zinc finger 0 assembles into a tetramer. This tetramer drives dimerization of this finger in a very stable structure. And the protein is now resistant to proteasomal degradation. So we have sort of an alternative view of this protein now. It looked like spaghetti before, but now we have a domain, essentially a new domain, this tetramer, within this unstructured protein. And we have an updated model of repression that's based on lots of functional studies I don't have time to describe, that actually say the tetramer itself is critical not only to stability of the protein, but the function of the protein as a repressor. And if you think about it, if you make enough mutations, you can make a monomer of this protein, at least something that won't form a tetramer. And we can do that, and it's not very functional. So we think this multivalent state is there to make a large structure which displaces activators from the promoter, helps to recruit the co-repressors, and ultimately repress the gamma gene. But from a standpoint of what does this all mean, we think we've identified opportunities now for direct targeting of this protein, which was, we thought, relatively untargetable before. You could interfere with formation of this tetramer, destabilize the tetramer. You might disable the dimer. We're exposing this degron sequence. Or you might target things to the surface of the tetramer, which is a very stable entity. And we're actually doing the last of these as a first option because we think it's the most direct. Shown here is what's a 3D printer version of the structure of the tetramer from X-ray crystallography. And you can see that there are pockets. There are regions where the subunits come together where there are pockets on this surface, which should be a region that we can put small molecules to at least get ligands to the protein. So we're currently, that's the current plan. We're establishing really the feasibility of this and trying to get through to show that we can actually target this protein in a preclinical sense, and then obviously we'll need to partner with the appropriate entities in order to really develop a drug. But the idea is to make a targeted drug for raising fetal hemoglobin. And that's sort of what I hope will be the next phase of this work as we go forward. I have a few final thoughts. One is focused mechanistic studies really can reveal novel and unforeseen biology and actually suggest opportunities for direct targeting of this protein, and I'm sure in many other cases as well. I think repressors have sort of a different mission than activators, and in this case, the repressor wants to make the biggest complex it can in order to really obscure the promoter region. And that's the goal of a repressor, is to be multivalent, to shut down the locus. And I think more broadly, there's an imperative now in the field for development of effective and safe therapies that are readily accessible to the many patients who get benefit. We need to go beyond the genetic therapies that are available if we can't make the genetic therapies fully accessible. Lots of people have contributed to this work. The early work on B11A to the sort of Achilles heel. We're dependent on these individuals here, particularly Vijay Sankaran and Dan Bauer and Jen Zhu. And the recent work is due to members of my laboratory shown here. I'll stop and I think we'll be ready to take questions.
And first of all, may I, on behalf of our audience and also on behalf of the university, would like to thank Shaw Price Rories for their wonderful presentation in this afternoon. And I think the presentations illustrated how important molecular science to the future medicine, which I think is very important to HKUST. As you all know, we are embark a journey to building our medical school. So now I would like to open the floor to have a few questions. I think we would like to take this great opportunity to discuss with these two distinguished scientists on the future medicine and also on the molecular science or any subjects related to the future development of medicine and biology. Okay, I would like to invite all our students, faculty members to have questions. Who will be the first? Okay, Professor Pu.
A quick question for Stuart. I think it's very exciting, the gene therapy. But my question is very general. Really, so for CAR T therapy, really they look at patients who, they're exhausted the other treatment methods. So because it's still, as you mentioned, it's ex vivo gene editing, it's still expensive. It poses some risk for the patient. For sickle cell patient or anemia patient, most patients are still managed by standard care, transfusion, and manage pain. For these approved, the FDA approved CRISPR therapy, how do they select patients who are good candidates for this, risky, somewhat risky, but expensive therapy?
Yes. So the question is, how are the patients selected, or who can receive the new editing therapy? Swee may have some comments, but in the trials, to my knowledge, and I was not involved in the trials, they have, first of all, they had an age cutoff. They have to be adults. They had to be severe. That is, they had many crises and hospitalizations and complications. They also had to have failed hydroxyurea, the standard of care, and they also could not have, I believe, matched bone marrow donors as well. So they were patients who didn't have many options. Now, once it's approved, especially with the effectiveness of the results, I suspect some of those criteria may be reduced over time. I think it's only approved now, at least in the U.S., for severe sickle cell disease, which is about a third of the patients that in the U.S. But I suspect over time, if the results look good, they'll relax some of those restrictions and also probably move to a somewhat younger age group, at least in trials, because I think the object should be to prevent complications, especially ones that are irreversible. So I think it's proceeding, I think, in a very cautious way.
Well, it is quite tricky. Quite clearly, as Stu says, you have to be quite selective. First, patients should really try hydroxyurea to see whether this can actually reduce the complications, and to really make sure that they're actually taking it. Because one thing that we need to be very careful is that patients get this romanticized idea that having genetic therapy could cure them of all the complications. So that said, so they have recurrent painful crises, and hydroxyurea doesn't seem to help them. But you also have to select those patients that do not already have too much organ damage, meaning, cardiovascular pulmonary complications, that they might not survive the actual chemotherapy that you need to clear the bone marrow. And then also to realize that if they have certain complications, the genetic therapy might not remove those complications. Like, say, if they have, kidney failure beyond a certain stage. So it is like choosing the right window and the right patient, which is tricky.
Next question. Oh, Nancy.
Well, first I want to thank Swee and Stuart for the wonderful talks. I think your talks really exemplifies the importance of leveraging basic research findings to the development of innovative therapeutics. So my question is that, during this process, there are critical steps that you need to ensure that it goes smoothly. For example, in Swee's case, you work with families who actually contribute to your research findings, and for the case of Stuart, how you work with the biotech companies to take it to the next step. So I just want to seek your advice, for us when we work with different research projects, when we encounter, these two critical steps. What kind of advice do you have? How do we, build the trust with the families, and also how do we convince the farmer that they should partner with us? Thank you.
Well, I think for me, I'm kind of a physician first, and winning the trust of the patients. Clearly, it is quite difficult because the patients with sickle cell disease, they're usually quite defensive, right? Because of the history of, which I won't go into. But having gained their trust and their confidence, and you can explain to them, now I have no problems recruiting patients to my clinical trials, the drug trials. But in the early stages, say when I was doing with thalassemia, I was just trying to explain to them that, this is to try and understand the genetics. And then, of course, it was much easier than, say, interventional trials, just like taking blood samples. But even then, you have to convince them that this was towards a good purpose, it might not benefit them. Actually, when you see those patients that those families have collected, I had to be convinced that there was something. And it's a question of just getting to the bottom of it. And I think sometimes, you might encounter a problem, but you know that it is there and you may not be able to solve it. You try to get around it, and sometimes you just put it aside until the right thing comes along. And I guess I was just quite fortunate at each stage that I could actually, crack the problem. Stu has your problems in a different aspect.
Yeah, I think I have my thoughts about biotech or pharma collaboration have changed, and it also depends on the situation. So first example is, although we were certainly willing to engage with companies once we had sort of worked out what we needed to do in terms of editing to, for example, raise fetal hemoglobin, we didn't at the time, which dial back almost more than 10 years now, there were a number of companies being formed around editing as a technology, but we didn't know which company was going to actually be able to execute. And what we did was, because you can put intellectual property around a sequence, we put intellectual property around the guide RNAs and made the license for that not exclusive, which meant that any company could take the license. If we'd made it exclusive, one company might have taken the license and either failed or sat, whatever. And I think that was the right decision. And fortunately, the company that first started CRISPR Therapeutics executed perfectly. And it's just, I think, good fortune for the field. There were several other companies that were involved at the same time, which did not execute well or didn't make the right decisions when they were at a decision point. So that sort of gave me one sense of the way to interact. More recently, when we're spending more time thinking about small molecules and companies, we've faced the barrier that the companies, each company has a different policy or attitude about collaborations, about ownership. And for example, there are some companies who are not willing to join with academics in a serious way because their business plan is to own everything. And that's obviously difficult. The other thing we face is that there are the people who fund us, particularly Howard Hughes Medical Institute, for example, has certain policies about what we can do as academics. And so we can't be working for a company if we have a collaboration, for example. So we have issues on both sides that we have to deal with. But I think where one can get the goals aligned, where we have the same goals as, for example, a small biotech or larger pharma, hopefully things can be worked out. Because the other realization I have is that to bring a therapy to approval requires the professionals, the ones in pharma or biotech. The editing therapy, for example, we have a bone marrow transplant program in our hospital. We could initiate the trials ourselves, but the investment in resources that a company can provide really makes all the difference. And I think the same is true in small molecules. I mean, we can do the proof of principle and show how to do it, but we need the companies to actually bring things to approval to make them drugs.
Thank you. Any other question? Yeah, please, lady.
Hello?
Over there. Yeah, okay. And afterwards, you.
Thank you very much for those amazing talks. I have a question on the drug itself, so the therapy itself. As mentioned in one of the previous slides, it is mentioned that sickle cell disease might cause small vessel occlusion, as there will be aggregation of red blood cells, sickle cells. And I wonder, after the use of these therapy, so there would be some, I would say, healthy hemoglobin, fetal hemoglobin there. However, the occlusions is caused by the previous adapt hemoglobin, and I wonder, would the effect, like acute pain or sometimes some tissue or organ effects caused by these occlusions, would that persist, or will you have some supporting or symptom targeted therapy or any methods to handle them?
Okay, if I understand you correctly, do these microvascular occlusion persist?
Yes.
Yes, and remain blocked?
Yes. Or will they be improved
Yeah.
by the new?
So, actually, the cells sickle and sickle all the time until they reach a stage where they are irreversible and they cannot unsickle. You see what I'm saying? And the idea is in the rate when they pass those very fine blood vessels is to be able to pass that blood vessels before the sickling kind of starts. So if you can reduce the rate of sickling, then they can pass it without having sickle and block the blood vessels. But when it finishes, the blockage will not remain permanent. The passage is open again. But there are instances where eventually it will cause damage to the endothelium, so you get, in fact, a form of what we call vasculopathy. And you can see that in very small blood vessels, like the bone ends, for instance. So they get permanent no oxygen supply. That's how you get what we call avascular necrosis in the bones. And the very fine blood vessels at the back of the eye also get blocked. And that's how they get this, we call sickle cell retinopathy. So some of these blockage kind of is stopped, and the fine blood vessels, you cannot restore the flow. Do I have answered your question?
I think maybe the other thing is you're asking what's reversible and what is not. Some tissue damage is irreversible, and some is reversible. And I think the key is to do the treatment before you get to the irreversible. Stroke is a major complication, sickle cell disease. Once you've had a major stroke, you can't reverse that. But the small vessel problem can be reversed.
The blockage is so, it does become unblocked. But with time, the damage to the organ will accumulate that we cannot reverse it anymore. So gene therapy, for instance, when they have eye damage, you can't reverse that.
Okay. Yeah, question.
Thank you for the very inspiring stories of how understanding the basic mechanisms behind disease and exploiting normal development can help to come up with a therapy. I think Dr. Orkin also raised an important issue, is about accessibility to the treatment and the ethical considerations regarding this, because actually probably the major populations who are affected actually are in Africa, for example, or in maybe even Southeast Asia. And the provision of this therapy from government sources is very unlikely in those countries, and maybe only for a privileged few in the US, and even under the NHS in the UK, it's extremely limited because it's so expensive. So how would you both think in terms of thinking going forwards in the, obviously, the cooperation between basic scientists and industry? On the other hand, industry wants to get back the money they've spent, and they want to make a profit. And on the other hand, to what extent do you think governments should actually fund this kind of research and taking it to therapy, which could possibly then make it more accessible to those who need it?
No, I agree. The therapy, unfortunately, the way it's currently delivered, cannot meet the need. We can't even do that in the United States. There are many places in the country where medical care or insurance won't cover therapy, or there's no insurance. My own view is that this is sort of a first-generation therapy, and we need to work at each step to try to make things more accessible. I think one would be to reduce the toxicity of bone marrow transplant. That'll certainly help. One could set up centers that could do this, do the therapy much less expensively than what's currently done. I suspect there are parts of the world where governments will invest in the therapy. For example, the Middle East is very interested in, they have lots of thalassemia and sickle cell disease as well. I think they could organize, and I think the governments are more likely to do it than, for example, the United States, where there are political issues to deal with. So I think each country is going to have to face it in a different way, but I agree that accessibility is key. One could say, you could take the other position, well, you shouldn't develop the therapy if it's not accessible. Well, we have to develop new approaches, and I think the editing or gene therapies is a new approach, and there'll be subsequent iterations to make it better.
But you're right. I mean, it costs $2.5 million a piece to do it. I mean, the NHS, even if they just can't sustain it. About 15,000 sort of patients sickle cell disease in the UK and top of Europe. So there will be only a handful of patients who can benefit from this treatment. So for instance, I'm focusing on developing new drugs. And there is one I'm working with a pharma that is very promising, and that probably could get FDA approval. And it has good anti-sickling properties. So this is for the way forward. But even then, with newly approved drugs, it still costs, what did they charge for Fetal? It was withdrawn, about $100,000 US dollars a year. So, as they often argue that with the gene therapies actually could be cheap in the long run.
My position is that the cost, $2.2 million, is a bargain. And the reason I say that is these individuals, many of them, the severe patients, let's say, with sickle cell disease, can't work. They don't hold a job. Their families are disrupted. The medical costs for a lifetime are estimated at roughly $1 million. And so the cost of curing somebody, particularly early, I think, it's a bargain, but I don't think that's the major limitation right now. The major limitation is delivery. You cannot deliver this therapy to thousands of people, irrespective of the cost. Even if it cost $100, we could not deliver it to thousands of patients. So I think cost is not the issue. I think it's not the place to focus.
Okay. I think it's now 5 o'clock. I have to bring this event to the end. And may I invite all of you to give the two presenters a big applause for themselves. And also, I would like to invite our President, Nancy Yip, to come to the stage to present the